Hello, everyone, and welcome back to the Radiology Advances podcast. Let's start straight away with one of the most, if not the most, difficult issue for clinical deployment of AI-based segmentation algorithms. Robustness in different scan conditions, scanners, and anatomies. Specifically, we're looking at, well, one of the more difficult organs to properly segment, the pancreas.
Exactly. The paper we're looking at today, Benchmarking Robustness of Automated CT Pancreas Segmentation, Achieving Human-Level Reliability Through Human-in-the-Loop Optimization, was recently published in Radiology Advances by Felipe Oviedo, Felipe Lopez Ramirez, Linda C. Chu, and their colleagues from the Johns Hopkins University and Microsoft Corporation.
Okay, let's start with the problem. What makes the segmentation of the pancreas so difficult? Deep learning models, they've gotten so good at automated organ segmentation.
Well, there's massive anatomical variability. Its shape is irregular and it sits right next to tissues like, fat, the gut, major vessels that can look almost identical on a CT scan.
So it's easy for an algorithm to get confused.
Very easy. So even the top models, they get these impressive mean scores, a high DICE score, for example, but they consistently stumble on the tough cases. It's what the authors of today's study addressed.
And the clinical consequences of that inconsistency, they're profound. You need accurate, consistent pancreas segmentation for everything, for quantitative biomarkers, for radiomics modeling, etc.
We're talking about high-stakes stuff like early cancer detection or risk stratification. If the segmentation is off, the entire analysis downstream is also off.
To be precise, multiple deep learning models achieve high DICE similarity coefficients, DSC, or normalized surface DICE, NSD scores, but still fail on a significant number of cases. And that's what brings us to the paper we're looking at today. How did Oviedo and colleagues approach this problem?
They set out to establish a new standard for defining what robustness actually means. DSC and NSD scores only capture the middle of performance curve. Those traditional metrics, they're fine for measuring overlap and boundary precision on average, but they tell you nothing about the variance. A high median score can easily hide, say, a 5% failure rate on the really complex cases.
And that inconsistency is what kills clinical trust.
Immediately. And what's more, most prior studies had one additional flaw. They benchmark their AI against a single human annotation,
which pretends there's one perfect ground truth.
Exactly. It completely ignores the fact that even human experts disagree with each other. There's an accepted range of performance. To really measure clinical value, you have to start by measuring that range. And that brings us to the metric they developed, the fractional threshold, or FT.
So how did they set that bar? What's the threshold?
So first they had to define what good enough means. They looked at how humans performed against each other, 4 different experts segmenting the same pancreases, and found the worst any human ever did. That became their floor. Only 1% of human attempts fell below that line.
And then FT just asks a simple question. Out of 100 cases, how many did the AI clear that bar? If FT is 0.95, that means 95 cases hit human level, but 5 didn't. Those 5 are your problem cases.
Exactly. A mean DICE score might look great at 0.88, but FT exposes whether you're failing unpredictably on a handful of patients, which in clinical terms is what actually matters.
So, the FT score tells you in a single number what fraction of predictions are clinically acceptable. It's a measure of consistency.
Precisely. It demands not just accuracy, but reliability.
So to test against such a high bar, their own study methodology had to be robust. They took over 900 venous phase CTs from healthy patients. They set aside 100 cases just for their test set.
Oh, absolutely. Right. And they focused on patients over 50, which is important because that better represents the population where you start seeing pancreatic diseases. And the test set is the heart of the whole study. Those 100 cases didn't just get one segmentation, they got 4 independent human segmentations. 4. 2 initial annotators, and then both of those were reviewed and finalized by 2 senior radiologists. That whole setup was absolutely essential to get a real reliable measure of inter-reader variability.
So before they even ran the AI, what did the human gold standard look like? How consistent were those expert radiologists with each other?
The consistency was remarkable, especially between the 2 final revised annotations. They hit a mean DICE score of 0.885, which is pretty high.
But what about the FT score, the proposed robustness metric?
That was the critical finding. When comparing human to human, FT hit 0.99 for both DICE and surface distance, meaning out of 100 cases, only 1 human segmentation fell below the threshold, a DICE of 0.80.
And this was healthy patients, so presumably the easier cases.
Right, this is the ceiling. If AI can't match humans here, it definitely won't on diseased pancreases.
Okay, so that's the bar. What happened when they ran the models?
To keep it short and simple, they evaluated three configurations. First, they trained four common neural network architectures from scratch. Second, they looked on pre-trained models, and finally fine-tuned those pre-trained models as a third configuration.
How did they do? Give me some quick insights.
The initial results were, well, they were exactly what you'd expect from the current state of the art. Their best model, a 3D UNet trained from scratch, got a mean dice of 0.88.
So on paper, that looks pretty comparable to the human average.
It does, but the robustness metric tells the real story. While the averages looked good, every single AI model showed a much higher standard deviation, a clear signal of inconsistency, and crucially, that best model's FT score was somewhere between 0.95 and 0.97.
Wait, so even their best model was still failing 3 to 5% of the time. So in 1 out of every 20 or 30 cases, this model could give you a segmentation that's clinically unusable. That's the difference between a cool research project and something you can actually rely on with a patient.
That's right, that 3 to 5%, that's the critical and clinically unacceptable gap. It separates that high average performance from true clinical reliability.
Okay, so the next obvious question is why? Why is that 3 to 5% failing rate? If the AI is matching the human average, what is it about the anatomy or the presentation that's tripping it up?
Yeah, and their systematic error analysis was so important here. They found basically four main categories of failure, and they all map to real-world clinical challenges. First, tissue contrast issues. Primarily, this was driven by fatty infiltration of the pancreas. When the pancreas undergoes fatty change, the tissue contrast just plummets and starts to blur into the surrounding retroperitoneal fat.
So the clear boundaries that the AI is looking for just disappear?
They disappear and the model gets lost. The second big cluster of errors was around anatomical context, especially in patients with a low body mass index.
Low BMI, why would that matter?
Well, in one case, the patient with the lowest BMI in the set, there just wasn't enough fat separating the organs. Those fat planes that help define the pancreatic margins weren't there, making it much harder to trace.
I see, so it's AI getting lost on the search for boundaries again. So what was the third category?
Poor boundary delineation. This happens all the time when you have, say, a collapsed duodenum right up against the pancreatic head.
An area where even humans can struggle.
Exactly, and the AI just couldn't reliably figure it out.
And finally, I'm guessing they saw the kind of errors that come from just not having seen enough weird cases during training?
You got it. These are the AI-only errors, things like misclassifying a piece of small bowel as pancreas or a big tortuous splenic artery, or a rare anatomical variation. They had a case of an annular pancreas, and the model failed. It suggests the model just hadn't generalized well enough to handle true outliers.
So if the problem is this inconsistency driven by these subtle changes in rare cases, and just training on more data didn't fully close that gap, the solution has to be smarter, right?
Yeah,
And that's where they introduced this human-in-the-loop active learning strategy.
this is their pragmatic solution for actually deploying something like this. The goal is simple. Let the AI do the fast, easy work, but get a human expert involved only where the risk of failure is highest.
Make the human's time incredibly efficient and sounds like AI as a co-pilot.
Precisely. And to do that, you need a really trustworthy red flag system, which they introduced as their acquisition function, Alpha.
Alpha. How did that work? How did it intelligently flag the bad predictions?
It's a really clever combination of two different signals of trouble. The first one is normalized entropy, which is a classic measure of model uncertainty.
So it's a way of asking the model how confident it is?
Kind of. It tells you what the model is sort of hedging its bets about whether a voxel belongs to the pancreas or not. But the second part is, I think, even more innovative, the complement of sphericity.
Sphericity. Okay, break that down for me. Why is that helpful?
Well, think about the shape of a pancreas. It's a characteristic, relatively smooth sort of bean-like structure. Sphericity measures how irregular or non-uniform the predicted shape is.
So if the AI spits out something fragmented or weirdly shaped?
Exactly. A healthy pancreas segmentation should be one smoothish blob. If the model outputs something jagged, disconnected, or anatomically bizarre, sphericity tanks and the alpha score goes up. Combined with high entropy, meaning the model itself was uncertain, you've got a flag.
And they didn't review everything, just the flagged cases?
Just the top 5%. 6 cases out of 100. After human correction of just those 6, FT hit 0.99, matching human performance. And the average time per case dropped from 35 minutes to about a minute and a half, a 23-fold workload reduction.
A massive reduction. And this is the crucial part, maintaining that human-level consistency and reliability. So when you pull it all together, what does this all mean for the future of clinical AI?
I think the big takeaway is that true clinical utility, it hinges completely on managing the exceptions, on reducing that performance variance. It's not about celebrating high averages anymore. For this stuff to get adopted, especially in high volume applications like screening, the models have to be consistent across all patients. And this hybrid model AI for speed on 94% of cases and targeted human triage for the hard ones, that's a practical, scalable roadmap.
And that is how we are coming to a close. Thank you for joining us for this Radiology Advances podcast on the study by Felipe Oviedo, Felipe Lopez Ramirez, and Linda C. Chu, and colleagues for benchmarking the robustness of automated CT pancreas segmentation and offering a concrete practical solution for clinical reliability. If you found this podcast informative, please rate, review, and subscribe. It helps more people discover our work and supports the broader conversation around innovation in imaging. You can read the full article and explore more great content on the journal website. This episode was generated with the help of AI, but all content was reviewed and verified by editor Dr. Luca Salhofer for accuracy.